Okay, good evening everyone, and welcome back to week 3 of MND-RA's State of Play information sessions. For those of you who haven't joined us previously, these sessions are replacing our MND Connect forum that we normally run every year. Obviously, due to COVID, it makes it difficult for us all to get together. So this year, we'll be bringing the research right into your lounge rooms and offices. Previous weeks, we've heard about how we can model disease in the laboratory. And last week, we heard about how we can improve our different models of care. Tonight, we'll hear from Dr. Mary-Louise Rogers from Flinders University in South Australia and Dr. Mehdi van der Bos from Westmead Hospital in Sydney. And tonight's subject is about how they're going to tell us about how their research can help us better measure disease progression in MND. Please feel free to ask questions throughout the presentation as they occur to you, either using the chat or the Q&A function if you're joining us through Zoom, or if you're on Facebook, you can use the chat function there. We'll be monitoring all those different forums to make sure we collect all your questions. And what we're going to do is have a joint Q&A session with both Mehdi and Mary-Louise at the end of the session. So that's pretty much enough for me. That's all you want to hear. And now I'll hand you over to Mary-Louise to kick us off for tonight. Thank you. Take it away, Mary-Louise.
Good evening to everyone, and thank you for joining us tonight, and thank you for Gethin for organising this State of Play. It's a wonderful thing. So today I'm going to be talking about measuring progression in motor neuron disease. So first of all, I'm going to just give a little bit of an outline of what motor neurons are, and a little bit of, not much, but about motor neuron disease and what's needed, and the significance of measuring progression and where to next. So if you just have a look at the this diagram we've got here, these little squiggles here are the cell bodies which are that control the motor neurons that firstly reside in the brain, in the motor cortex, and they send signals down to the cell bodies that reside in the brainstem and the spinal cord, and they project out to limb and breathing muscles and other muscles that control breathing and speech and swallowing, and these are the actual neurons, these motor neurons that are affected in motor neuron disease. So the disease is actually characterised by a progressive limb weakness, respiratory issues, stiff muscles, and I'll just move this. Just hold on a minute. Just having trouble seeing my slides. Let's go back. So people report a progressive limb weakness, respiratory issues, stiff muscles, hyperactive reflexes, and then impaired swallowing and speech. And this proceeds to end-stage respiratory failure. So what do we know about the disease? What we do know is there's a lot of heterogeneity, and what I mean is a lot of variability in the disease. So some people present with weakness in their limbs, and some with problems with speech and swallowing. Some have some dementia. There's a high prevalence in males versus females, and Caucasians have a higher prevalence. And the age of disease onset, there's a high prevalence of around 60 years old. And 10% of people have a familial, that is, its cause runs in families, but most people don't. And so what we know is there's defects that happen. What happens in when you get motor neuron disease, you get neurodegeneration, progressive neurodegeneration, and you can see when they look in the cells, they can see defects in protein processing, mitochondrial dysfunction, nerves, cell transport, DNA, RNA processing. You've probably heard of all these terms, and these are just some diagrams. This is a motor neuron, and these are these support cells. So the other thing that's known about MND is that it's actually probably a multi-step process. That is, there's maybe 6 genetic or environmental exposures that may trigger the disease. And interestingly enough, a study in the UK conducted by Amar Chalabi, who's at King's College, showed in Europe there's 6 steps that probably are needed before people actually present with the disease. And Steve Vukic, who Mehdi works under, he also found this in Australia, Korea, and Japan. So there's some sort of process that happens in the disease. So, but unfortunately, there's no treatment there's no cure and limited treatment. So both drugs that are used, riluzole and edaravone, can only improve outcome for a few months. And there's many clinical trials have been tried and a lot of failures, but what really is needed you really need to understand the variability. Why is this, there's a variability across people, and maybe we can redefine those groups that may be more similar so that we can help assist in clinical trials. We can do this through biomarkers. So what I mean by biomarker is this is a measurable characteristic that indicates biological, pathological, or pharmacological responses to a therapeutic. That's just that's a definition that's sort of accepted across the world. They actually provide accurate indications and quantify biological processes. There's a number of biomarkers, and some of these I will talk about. Progression biomarker, prognostic marker, and pharmacodynamic marker. Diagnostic, when I'm not talking about that, because until we have a cure or a drug, I think diagnostic biomarkers are not necessarily needed. So what they can do is actually help define subgroups and help define people that can respond better to clinical trials. So reducing the variability. There's lots of heterogeneity, which I talked about. There's clinical, genetic, demographic. So can we reduce the heterogeneity by getting biomarkers that can work in prognostic, that is, define subgroups of people that can respond to treatments and enrich the trials. So there's lots, when I'm talking about trials, there's lots of clinical trials, and just to show you a few that's been ongoing or finished in Australia, Tessa-Vadera, and actually we've been measuring outcomes in that trial, and part of that was also sponsored by MD Australia, and as well as TriMech, which there's 2 phase 2 and phase 3, and I'll talk about that in a minute. So we are, I'll be talking about the biomarker P75. So there's a number of clinical trials, but really we need good biomarkers. So, and they will reduce variability for those that are taking part in the trials. So what I mean by biomarkers of progression, these are ones that change over time and that can be measured. Currently, the only validated clinical outcome is the ALS-FRS. This is the revised amyotrophic lateral sclerosis, another name for MND, functional rating scale. It's often people can phone in about this questionnaire based. There's 12 questions, and they're often asked every 3 months. So there's these 12 questions here around speech, swallowing, dressing, walking, how short of breath, and respiratory function. And so, for example, climbing stairs goes from can't do it to normal. So 48, so 4 would be normal, and so you can see that it is quite powerful, but it still is subjective. So it would be good to have something biochemical that we can, or physiological, that we can measure that changes over time. So there have been imaging biomarkers that have been proposed, and neurophysiological, which Dr. Mehdi Van Den Bos will actually give a talk about this next. And there's been numerous searches for biochemical progression markers. Neurofilament, which is these substances that are released by nerves, have actually been extensively shown to be quite good prognostic, but not necessarily progression markers. And the marker that we've been looking at, which is found in urine, is called P75 extracellular domain. I'll be talking about now. So just looking at this diagram, all I want you to take away from this is this red squiggle here. What happens when a cell gets sick? A part of this molecule gets cut off, and it's called an extracellular domain of this thing called a receptor, and it gets, as the cell gets sick, it gets cut off, and it goes to the blood and then to the urine. So we've been looking at this marker called urinary P75 extracellular domain because it's actually quite high during fetal development in expressed on the surface of motor neurons and other cells, but it's not normally expressed in healthy individuals. But what it does is actually get re-expressed, and it's shed. As I said, there's little parts of, so a part of this whole molecule called the extracellular domain is shed. And urine is a good source of biomarkers compared to cerebral spinal fluid, because it's much easier to get than taking a spinal tap. So what we've done is design an enzyme-linked immunoassay. And what we do is we have these things called antibodies, and we have urine and we actually measure biocolor reaction. We can quantitatively measure this P75. And what we've shown is that, see all these little red dots, these are the P75, and this is in healthy individuals, and this is in people that have MND. And you can see that there's more in people that have MND compared to healthy individuals. And this is taken at the first visit to a neurologist. So the other thing that it was actually correlated to that ALS FRS, this measure that declines as people lose more and more function. So it does indicate health status. So what we did is do a longitudinal study and asked whether it changed over progression, because that's what we were really interested to find, whether it was a progression marker. And so it was run out of the US and Australia, and most people had, so there was around the age of around 63, and we had around 31 people that had at least 3 3 times that they were collected over 2 years. This is over a 2-year study, so every 3 months. And so what I can show you here is that, see these red dots here, and the lines are individual patients. So what we showed was that P75 did change over progression, whether it's from when people were diagnosed or when they had their symptoms they reported to the doctor, or from baseline, which is the first visit to the neurologist. What about prognosis? Prognostic can actually help us define different subgroups of patients to help reduce this heterogeneity. So we know that neurofilament light is quite a very good marker of prognosis. But what we published a couple of years ago was P75 is also a prognostic marker. So what this is called a Kaplan-Meier analysis, and it just shows that those people that had higher amounts of P75 at their first visit to a neurologist had a shorter survival than those people that had lower amounts. So it means that it can predict prognostic, or how long people can survive after baseline visit. So what we're interested in is pharmacodynamic marker as well, and whether this marker changes over progression and with a treatment. And in this case, we looked at a treatment, which I said before, which is this antiretroviral treatment, Trimec. And this study was, this was actually published last year, and this is called the Lighthouse Trial. And what it showed is that it was tolerable in 40 people, that it was tolerable. And we did measure P75 levels, but basically what they were looking at is people get this HERV-K, which is like a retrovirus that everybody has, and we think it's reactivated in some people that have motor neuron disease, and this treatment actually reduced that. And people seem to be, this ALS-FRS is this is normal, and this is the normal projection, and they seem to be having less. This is not a trial to show that the treatment is working. It's just a trial to show that it was tolerated, but it did seem to say that we should go to the phase 3. And what the P75 showed, where you can see is high here and it's low here after 34 weeks on the treatment. It's actually 28 weeks because I started here, was that there was less than expected P75 in that we would have expected. So it seems to be, it may be beneficial, we don't know yet. So it's going to a larger trial, which I talked about before. So what we really also need to do is, so what I've told you is P75 is maybe a quite good marker of progression, and also could be used in clinical trials. But what about other urinary biomarkers? So we're looking at this other marker called neopterin. So this is actually present in a small inert molecule, and it's released as part of when people have a immune cell activation, or they have, and that also happens with motor neuron disease. So we wanted to ask some questions. This is funded by M&D Australia. We wanted to ask whether neopterin differed between people that have M&D and healthy individuals, and how did it compare to this urinary P75, and can it be prognostic, and does it change over disease? So like the other P75, what the other work that we did, we actually used this enzyme-linked immunoassay. It was a little bit different. It was a more simple assay because it's a very small molecule. So I'm just saying that we're able to measure it. And what we've done is look at, we had a 24 healthy individuals and 43 people with M&D, and we started and measured the neopterin at baseline to start with, and that is the first visit to neurologists. And what we found, there was a higher level in those people that have M&D, and this is in comparison to the same people were measured with P75. And very interestingly, there was a correlation between this neopterin and the P75. So, so the neopterin's level measuring the P75 is and the neopterin are correlated. So what we also wanted to know was whether there was whether it was correlated with the disease that's measured by the ALS-FRS, so disease state. And what was very interesting, those people that were younger than the median age of 67, there was a very strong correlation between neopterin and ALS-FRS, but there wasn't over 67. So it seems to be a marker that shows immune activation, but only if those people that are under a certain age. And but in normal people, there was no such there was no significant difference over age. So we also looked at this prognostic value, and like P75, neopterin was also prognostic. So the higher amount, this is a median amount of neopterin versus a lower amount. And what if you have a higher amount of, the higher amount of, this the median neopterin was 141 millimole. And what happened is if you had a higher level than that at your first visit to a neurologist, then you had a shorter survival. It's sort of similar to what happens with the P75. So we also were able to show that in similar ways to P75, there was a change in progression, in 22 individuals. So it seems to be that it changes over disease. We haven't got to the stage where we're measuring rate, but it does change over disease progression. So I just want to summarize this. So it's significantly, neopterin was elevated. It's correlated to disease state in MND. It's actually higher in those, but only in those people that are younger than 67. And it's correlated to P75. So the immune status is correlated to this neurological marker, which is quite interesting. So what we've been able to show is that urine is a great, so it's a great fluid to look for biomarkers, and our lab is actually pioneering biomarkers that can be used in clinical trials. So we really are going to be looking at neopterin in a number of clinical trials to see whether we can improve clinical trials by subgrouping people according to their neopterin and P75 and together. And we're also looking at other markers. So I'd just like to thank everybody and thank MND Australia and also all the people in my lab that did this work, Vasilis Koronis and Stephanie Shepherd, who's back now. She was a student in my lab and went to the UK to work at Sheffield and has come back to our lab. And David Schultz in the fourth generation clinic, and my collaborators overseas, Michael Benatar and Andrew Malaspina, and all of the neurologists in Australia, and most of all the patients and participants, and Ian Davis, who actually has been from Fight MND, that actually has sponsored our move to a high throughput screen measurement for P75, and MND-RIA and all the funding bodies that we've received. Thanks very much. Should we go back?
Okay, great. Thank you for that, Mary-Louise. And now we'll be hearing from Mehdi van der Bos, who's going to talk about a different way of looking at disease progression. Thank you, Mehdi.
Great. All seems to be working well, I think. Is that the case on your end, Getin? Okay. Now,
it looks good. You just need to put it in presentation mode. Yep, slideshow, whatever.
All right, sweet.
Cool, looking good.
Thanks very much for inviting me to be part of the panel today. So I'm a neurologist and I work with a team of different neurologists, and we work in Sydney at Westmead Hospital, and we're all skilled in neurophysiology, and we're interested in why does this horrible disease essentially develop and progress, and how we might better assess that and work out a way to use that assessment to understand and deliver a cure. So you know that ALS essentially starts with focal weakness in one area, and it progresses to involve all regions of the body. And in this way, it ultimately takes the lives of at least 2 Australians each day. There's a few different ideas competing as to its mechanism, and there's increasing evidence that a cortical origin for the brain is actually a reasonable idea, with cortical or neuronal excitotoxicity leading to death of the cell through excitotoxic death. And this river of excitability may start in the brain and cascade down the upper and lower motor neuron, as you can see over here on the right, to result in degeneration of the upper and lower motor neuron. So, as Mary has mentioned, ALS has been proposed to be a multi-step kind of process with multiple different steps needed, much like cancer models. And this kind of idea was developed from modeling of data from incidence in different populations, and it's valid seemingly across different parts of the world. And people with genetic mutations may have less steps, but they all end up in the same place with this devastating disease. And perhaps importantly, there's actually models that suggest, or ideas that, from eminent neurologists that have suggested that a lot of these changes may start a long time back, and a mixture of genetic and kind of environmental factors act on each of us, who are also different in ways that produce the sickness of motor neuron disease. And this sickness may be there for a long time, even perhaps before the actual development of symptoms themselves. And then the progression from symptoms of focal weakness to spread throughout the region and ultimately take someone's life is a long process. But again, this idea of a long preclinical period has some merit. Now, what does neurophysiology do? So physiology, and neurophysiology in particular, provides insight into the function of systems, and in this case, neurological systems. Neurophysiology provides an insight. Really, it's just a tool that helps us understand the workings of a biological system. And so in understanding how neurophysiology might work in a person who's healthy or has a different type of disease, and then contrasting that with ALS, we understand something about the pathophysiology of ALS and how the disease progresses. Understanding that pathophysiology is really critical to delivering a cure ultimately. So how can we reliably measure ALS disease progression? Well, there's different ways of doing that with neurophysiology, and that can guide clinical care, but perhaps at the moment of most interest is the acceleration of the search for a cure, and the use of biomarkers in early phases of a trial or testing of a drug, really, to improve the sensitivity and improve the ability to pick up the right drug, and as Mary-Louise has already described. We've all seen this muscle wasting, which is the hallmark of ALS, and it can start small in this kind of classical presentation of ALS on the left hand with a left-hand side with a split hand, to the more generalized wasting and weakness that we have unfortunately recognized. This is a manifestation of disease within the lower motor neuron. Now, electricity is used by our body to transmit signals. And that electrical activation can be used to study the lower motor neuron. You might be familiar, actually, with the nerve conduction study, which allows us to use electricity to activate the peripheral nerve and record the resultant muscle twitch with surface electrodes as a bipolar kind of, it depends on the actual nerve, but as a little electrical potential. And that can tell us the kind of speed and size of the muscle response. And actually, when we stimulate a nerve, there's two impulses. One, the immediate muscle twitch that you see, and electrical current goes both ways because the nerve's excitable in both directions. And it transmits all the way up to the spine and then is reflected as a small little, from the spinal motor neuron pool, as a small little blip known as the F wave, which we can record, maybe 20 milliseconds later on, depending on the nerve. Now, people have studied this very simple response and actually devised one of the more reliable neurophysiological biomarkers called the Neurophysiological Index, and where they take this kind of muscle twitch and measure the size and then the DML, or the distal motor latency, so the time it takes from the wrist stage to the hand to develop that twitch, and then also to see how many of these smaller reflections from the pool, the spinal motor neuron pool, the F waves, can come back. And people have demonstrated, clever people have demonstrated that what happens in ALS is obviously we see the muscle getting smaller. So the neurophysiological index will reduce. The distal motor latency will actually subtly get longer. And the F waves will become less prominent. Actually, as you can see on the right hand, what happens with the F waves is that these responses, because there's less and less motor nerves left over, these responses become a bit, stereotypical. So the same ones come again and again with the same latency. And initially they're a bit bigger because of a variety of reasons, but essentially the motor units that are left over are a bit bigger. And as the disease progresses, these stereotype responses become less and less frequent as well, really leading to a decrease in persistence. And that's recognized as a change in the neurophysiological index. There's other ways to look at these lower motor neuron estimates. So for example, the surface potential really captures an overall strength of the muscle, but we can break that down by doing different types of stimulation and get an estimate of the number of motor units within that bigger surface potential. And as you might know, with motor neuron disease and degeneration of a motor nerve, the remaining nerve fibers or the motor units that are left compensate, that become bigger. So superficially, the muscle can look the same. But when you probe a bit deeper, you'll see that that big response is made up of smaller, it's made up of fewer responses, each of them being bigger than before. So you can see this kind of incremental change there. There are other ways of probing the lower motor neuron. So there's this electrical impedance myography, which uses applies some electrodes and uses high frequency currents and measures kind of resulting voltages and changes in them through other electrodes to look at the changes in the structure and composition of muscle, and in that way it can pick up degeneration and hypertrophy of muscle, and as well as fatty replacement, and that's been demonstrated to be a useful biomarker as well. Now, the area that I'm actually interested in is more to do with more complex cortical neurophysiology and this idea of the origins of the ALS residing in the brain. So the upper motor neuron has actually been very difficult for neurologists until recently to assess reliably, confined in some sense to many of the simple reflexes of the bedside examination. But the technique of TMS essentially opened up the possibility of probing the brain's connections to the motor system in a unique way. So we use a magnetic pulse to activate the cells of the motor cortex, the interneurons and pyramidal cells, the pyramidal likely representing the upper motor neuron cells. Now, we use different types of stimulation parameters, different timings, strengths of different pulses to select populations of interest within the motor cortex. And that tells us about the projections onto the upper motor neuron and the function of the motor cortex in that area. We can use that magnetic pulse, and what it does is activate these populations of nerves in the brain. And over the motor cortex, we can generate a cascade of volleys that go down the upper motor neuron and connecting the spine to the lower motor neuron. And seen as a muscle twitch, much like the electrical activation of the wrist might be, but it's had its origins all the way in the brain. And that muscle twitch is a similar motor recorded with surface electrodes and is known as a motor evoked potential. So early work using TMS had mixed different signals. And there's this technique of threshold tracking TMS, which Professors Vucic and Kiernan, who are based in Sydney, helped develop and nurture and carefully deployed to the study of ALS. And they've really done dramatic things, or great things in terms of uncovering the abnormalities in the cortex in ALS patients at an early stage. One of the earlier kind of measures that they devised was this phenomena, or helped implement, I should say, it was this phenomena of short interval intracortical inhibition. And what they demonstrated was that there were these little circuits in the brain, in the motor cortex, that were kind of produced by inhibitory interneuronal cells, GABAergic interneuronal cells, that were abnormal in ALS. In fact, they were dysfunctional. And in ALS, in ALS patients presenting early, you could capture this abnormality with a change in the potentials needed to generate them. So these potentials were weak, and so that was represented as a reduced threshold. And these reduced thresholds in people with ALS were demonstrated early on in the disease course. This was used in lots of different ways, but in terms of the things that we're interested in today, what we found was that this abnormality in ALS, people had demonstrated that when you take riluzole, one of the few treatments that we have available, this kind of inhibition was restored. So one of my colleagues actually studied this, and he said, well, he found that the changes in the cortex, which could be demonstrated with TMS, when people were commenced on riluzole, they kind of normalized a little bit, and that normalization lasted a similar length of time to the changes that we found, that we found in terms of patient survival. So that demonstrated a kind of a biological basis for the mechanism by which riluzole worked or helped to support. There's other implications from this technique, and one of them is actually pretty important. It's actually improving the diagnostic window. One of the things that's difficult with ALS is that by the time people have gone through a sieve of different, clinicians from GPs to neurologists, it's hard to, to diagnose the disease early. And ALS itself may well have a long preclinical period. The use of TMS has provided for an earlier window to diagnose ALS. And that's, with high sensitivity and specificity, can bring forward the diagnosis by as much as 8 months, which is a lot. This early kind of study of inhibition, though, didn't kind of directly demonstrate this cortical hyperexcitability. So a study we did earlier probed a different set of neuronal circuits that had been previously very hard to study. And we used the threshold tracking technique to demonstrate this phenomenon of SICF, it's called, the short interval intracortical facilitation. And it's this kind of phenomena which is abnormal in people with ALS. In particular, it's increased in people with ALS. So they have more facilitation, more hyperexcitability. And what we demonstrated was that the cortical hyperexcitability is actually a paired shift, represented by a paired shift in inhibition and facilitatory circuits, so that people with ALS had less inhibition and more excitation, as demonstrated on the right-hand side. So if you look at this entire span of threshold change, there's more inhibition in the span, more facilitation and less inhibition. And in people who are healthy, who are older and age-matched, have less excitation or facilitation or SICF, and more of the inhibition. Now, you have to excuse that somehow the slides are going forward in automatic timing, but hopefully I'll pull it back just as well in time. Now, this kind of index of excitation that I demonstrated here on the right-hand side, is that biologically relevant? Well, it is. People with a lot of more disability, as measured by the index Mary-Louise has mentioned, the ALS-FRS, these people had increasing disability. They had more symptoms when they had a higher index of excitation. And there's other measures where that might be a bit more relevant. Now, another question that we're trying to address is, how does the disease spread? And one of the potential mechanisms, for example, if the disease starts on the right hand, how does it spread to the left hand, are the pathways that connect the brain, the kind of interhemispheric conduits. And that is kind of, these pathways are kind of through this body of fat fibers called the corpus callosum that links the hemispheres of the brain. So the transcallosal pathways might be a potential conduit for the disease's spread. One way you can kind of think about them is that they present inhibitory signals between the sides of the brain, and they allow kind of damping down a phenomena between hemispheres. That's one simple way you can think about it. And we can measure that inhibitory signal between the brains using the technique of threshold tracking, again, by stimulating one hemisphere, recording the muscle twitch in the other hemisphere. And at the same time, that stimulation with just the right parameters can trigger inhibition of the other hemisphere, which we can also measure by a change in the strength of the muscle twitch. What we demonstrated was that people with ALS had reduced inhibition as measured by a change in their thresholds. And they had that in both hemispheres when compared to healthy older controls. And these are just averages of all these different stimulation parameters to give you this neat kind of little average of the results in each hemisphere of ALS patients when compared to healthy older controls. Now, transcallosal dysfunction, when you kind of think about it, you might want to kind of generate a measure of the changes in inhibition across both hemispheres and see how that might relate to disease and disease progression. So we summed the hemispheric inhibition across both hemispheres because it's one body, so it's more naturally representative of changes across both pathways. And what we demonstrated was people with more inhibition, or less inhibition really is probably the most interesting way to say it, less inhibition tended to have a more rapid course of disease progression. Now, are these measures really kind of relevant in a longitudinal study? Well, there's cross-sectional approaches to assessing disease progression, as I had mentioned before. So, for example, the graph before takes the time people have had symptoms by the time, time people have had symptoms at the time when they first meet the neurologist, and looks at kind of how many points that they've lost across this period of time. That's one cross-sectional way to kind of define a rate of change. Another is just to study people across a period of time. And one of my colleagues and our team, really, because it's quite a difficult effort to study lots of people across time, studied, used TMS across many different people across a year-long period. And what she's demonstrated is this phenomena of SICI that was first optimized by Professors Keenan and Vujic in ALS really decreased across the period, or a year-long period of study, quite substantially. Now, people have applied this to the study of drugs, much as Mary-Louise has already described for the urinary markers, and demonstrated changes there too. For example, as this study with this new drug called retigapine was studied with neurophysiological parameters. So it's a phase 2 trial that was recently published in the US. And what they demonstrated was that markers such as SICI were actually improved and increased about 6 to 8 weeks after the initiation. It was really at 8 weeks in the high dose group after the initiation of treatment. And so, I mean, it's a phase 2 trial, and what it does is look whether there's a biological marker that's of relevance that can maybe be used to screen that drug and see if it would be suitable for a bigger trial. And it does look like that's a useful biological result. Another kind of a different approach to looking at biological or biomarkers to accelerate drug trials, or measuring ALS disease progression to accelerate drug trials, was proposed by colleagues in the US, Professors Rutkov and his colleagues at Harvard, had looked and thought, look, there's all these different noisy signals that we can measure, but if we measure them more frequently, maybe it can more reliably track the course of disease change. And so they took a bunch of different measures, some not so simple, such as electrical impedance myography, and others more simple, like the vital capacity. There are other papers which describe speech and other sorts of phenomena, and things like hand grip. And what they demonstrated was if you measure something frequently, you have a more precise measurement of the trajectory of the change in the disease. Now, some markers are more useful than others to capture the significance of biological kind of change in a person. But all of these measures were improved by the frequency of measurement. And that's perhaps an interesting idea that will need to be incorporated in other approaches to biomarkers in the long term to improve that sensitivity and ability to capture signal in what is a relatively rare disease. So neurophysiological measures in ALS, they can provide insight into the pathophysiology of the disease's progression. And there are these features of cortical hyperexcitability and transcallosal inhibition, which are both present early in the disease course, appear to track the kind of disease morbidity and rate of progression, and may be useful in disease trials. The thoughtful use of ALS biomarkers has the potential to dramatically accelerate the search for a cure. Again, I'd like to thank the MND Research Australia for inviting me tonight, and also for our support in our research, including the Beryl Bailey Fellowship. I'd also like to thank my colleagues, Professors Vucic and Kiernan, and our other colleagues, together with the University of Sydney and the New South Wales Health. Thank you very much for your time. I think we're moving on to questions shortly with Getten moderating those, so I'll stop my screen share and allow him to transition on.
Great, thank you, Mary-Louise, and thank you, Mehdi, for some fascinating insights into the world of measuring MND. We've got a few questions coming in from the audience, so I'll start you up. Mary-Louise, you're up first. There was a question that's said in one of your early slides, you mentioned a type of MND that was not true MND. Could you just give us a bit more information on that?
So what I was talking about is that some people may have just symptoms that mean that they have a problem with upper motor neuron, or some that have lower motor neuron dysfunction, but that's not true MND. And it's often what happens, and Mehdi will be able to tell you about that, is it's often shown over time. So some neurologists may think some people will have MND, but actually if it progresses very slowly, so this progression, which is what Mehdi and I are measuring, if that's, over years instead of months, and they have a pure upper motor neuron disease or a pure lower motor neuron disease, rather than the true motor neuron disease that means that you have both upper and lower motor neuron dysfunction and progression in both of those types.
Mehdi, would you like to elaborate a bit further from a neurologist's perspective?
I guess there's a few different perspectives on this, but essentially there is a lot of variability in people, and that's, reflective of the different nature we each are. And there are distinct disease types. So there's PLS, PMA, and there's other types of diseases we might consider within the MND umbrella, like bulbar palsy or shooter, like all these different sorts of things, which are not quite the same as this common variety that we think of as amyotrophic lateral sclerosis, this mix of upper and lower motor neuron degeneration. And I think all of the research aims to find a cure for this kind of cascading disease. But essentially, so getting back to the original question, really, I'm not sure that, it's really, they're kind of academic points of different types of distinction in our struggle to understand the disease. But the reality is they each tell us important things about the diseases, motor neuron diseases. And it might be difficult, though, in each of these, with some of these biomarker measures that we measure, for example, it can be sometimes hard to capture the variability of the spectrum of some of the more rare phenotypes. Does that make sense? So if we've got a marker, for example, that really is strong for the upper motor neuron, captures none of the lower motor neuron, well, it's going to be hard to capture that disease phenotype. Yeah. Having understood that, though, some people with ALS progress slowly. Traditional ALS to just progress slowly. Why does that happen? And other people progress quite rapidly. Within a few months, they've passed on. So understanding that they're distinct problems, I think, is probably the easier way to think about it. But understanding there's variability in people, and then there's the disease subtypes, of which, they're all, uncommon. They're not the day-to-day thing you see walking in every day in the neurology clinic. But, they're common enough that many of us, or many people, will suffer from it. So a different way of thinking about it. There's much variability in ALS, but there are also distinct subgroups within motor neuron disease.
Okay, great, thank you. A question we have here, which probably goes to both of you as well, that one of our audience members are very impressed with the trials we've had to try and determine the effective biomarkers. The question remains, and I think Mary-Louise touched on this, is while an early indication diagnosis is welcome, if there is no cure at this point, how does this help our patients? Or are the biomarkers useful in determining better pathways to finding drugs that will help treat the condition? Mary-Louise, if you want to go first.
Yeah, so really it's exactly what Gethin said, is that what we really need is something that can help us determine effective treatments, and so progression markers, the urinary ones and the neurophysiological ones that Mehdi has described. So you could detect the treatment that works, and so rather than diagnosing. So it's much more important that we find a treatment than if there's no effective treatment, then it's really not going to help people being diagnosed earlier. It's going to help people if we find a treatment that works. So we're trying to find something that, for example, reduces the urinary biomarkers we're looking at. So, and that would indicate that that treatment is working or not. For example, the Triamec, or something that affects the immune, like a drug that works with the immune system. We can, for example, in the future, look at Neoptrin and see whether the levels change if the drug is working. Just an indication that the drug is working is maybe a good way to describe it. So if it's working, then we can hopefully find these new cures through using these biomarkers.
And I guess that by having the biomarkers, they're much faster than waiting for actual disease progression changes as well, yes. Which is the whole point of biomarkers, basically. They're just a bit faster. Yes, Mehdi, you're...
I'm going to throw a slightly different perspective. So if you just think about it, biomarkers, all of these things are tools. We don't need to be religious about any devotion to one tool or the other. They're just tools for a job. If we look at them from the perspective of a trial, we'd like to accelerate the ability of a trial to find a cure. Okay, that's without doubt a true thing. The reality is there are a lot of diseases which have neurodegeneration involved, which have been up against this for, with larger funding for longer. And a common thread amongst these is that the question of whether the disease starts even earlier. So this is the case in different types of things like Alzheimer's and other sorts of things. And the reality is that if you want to find, if the disease starts earlier and our treatments are kind of given at this point where they're no longer effectual, can't really change the clinical course in any meaningful way. For example, if I have Alzheimer's dementia and I'm demented and unable to look after myself, arresting the point of the disease and leaving me in this kind of state where I'm perpetually unable to look after myself and relatively not so well, makes it difficult. For the same reason, a lot of clinical trials, if you'll notice, are focused on the early point of the disease, because that's thought to be when it's the best opportunity to arrest the clinical course of the disease. So if we only aim to find a biomarker and don't have things that push forward the envelope at which we can diagnose, we actually may make it harder for us to ultimately successfully identify a cure, because we might end up just looking at a point when symptoms are obvious. Another way of thinking about it is we're understanding ALS is more and more a whole brain disease. And there's a myriad of different ways that can be manifest. And part of them are through the cognitive pathways. There's often other cognitive changes in people that sometimes may herald the onset of a neurodegenerative disease in different people. And that ALS is one of those potential areas where that might be the case. So I think if we just focus on, we need a biomarker to kind of accelerate drug trials, we need to have specific, goals for that drug trial that are married to the biomarker. So whereas if we kind of if we only look at biomarkers and discount any kind of exploration of their utility in diagnosis, then we lose the opportunity to bring forward that therapeutic window to a point where people aren't ravaged by disease in all four limbs. They actually might just have a bit of mild weakness. So perhaps, ideally, to be frank, we might pick up some herald of cognitive dysfunction and corroborate that with a kind of a biological biomarker or a neurophysiological marker, whatever it happens to be, and arrest the point of disease before they have cognitive or motor dysfunction. So it's a different way of thinking about it, perhaps a complementary way.
No, I actually agree with you. I think it's actually a really important point to make, and just to re-emphasize that there's been, a number of studies that have shown that, for example, the use of neurofilament, which does rise very early in the disease, and those people that have a genetic form of the disease may be one way to make a trial very homogeneous. So you've got them early, the gene that causes that disease, and there may be a treatment, and that's some of these trials that are actually are ongoing at the moment, where they're trying to ameliorate the genetic forms. That's exactly what they've been doing.
Actually, just a slightly different kind of concept. If you think about cancer, I mean, that's like a biological model that lots of people have understood through science, and in the audience might understand through science and even high school teaching. Like, that's a multi-step process. There's biological markers at each of these stops. The cancer might be localized or cut it out. There's kind of this particular cancer might spread more through the blood vessels because of these biochemical changes. There's these biomarkers. We'll target it like this. ALS, with its modeling, looks to be a multi-step disease. So having a biomarker and understanding of the disease better at each of these steps might allow us for interventions strategically or in combination, as it is in cancer and other diseases, at earlier stages or more effectively through a combination of, as Mary was saying, these understanding of these unique and even tailored, perhaps, treatments for the patient suffering.
Just a slight aside to that. So I think there was a recent study on riluzole that said that basically the earlier patients get treated with riluzole, then the more effective the treatment is. So I'm guessing there that having better biomarkers to give you a more definite diagnosis as early as possible in disease is going to help there, even when we have only limited treatment options at the moment.
That's a fair enough point, yeah.
One more question we have here is, this is for Mehdi. Could you explain how oxidative stress may lead to MND?
Fair enough. Do you know what? That's probably a better question for Mary-Louise, because she's some biochemical background, to be honest. But my simplistic understanding is, I mean, in terms of the detailed instructions, my simplistic understanding is that essentially oxidative stress can be one of the exogenous factors that leads to neuronal cell death through excitotoxic mechanisms, which is that excitotoxicity is that kind of final cascade. Oxidative stress can have an impact on a range of different things, from our DNA to kind of cellular function and a range of different kind of pathways. And there's genetic mutations which might predispose us to increased oxidative stress. But I think Mary-Louise will probably have more detail to kind of.
I think you've explained it quite well, because it's a complex process, and so edaverone is supposed to help with this oxidative stress, and this is, but in fact it only works on a very small subgroup, of people with MND. So that's quite interesting, and those people that are quite fast progressing. So yeah, so no, you've explained it well. It's just one of the many pathological processes that have been identified that happen in MND, and all of these processes we've come to know because we've found all these in the last 20 years through finding a lot of these genes that cause 10% of MND, which is a great leap forward, I think.
Thank you. We've just got a final question coming in now from Duncan, and a very astute observation. So the first question is, could gut biota end up being a biomarker, do you think?
Look, I think that's an emerging area of research. I mean, again, Mary-Louise would be a great person maybe to comment on that more. But at the end of the day, like, there's a whole plethora of research really exploding over the last 5 to 10 years in gut biota and our understanding. And it's not that easy to tease it all apart, to be honest. So it's easy to find statistical significance in lots of things when you kind of just cast the dice many times. I think there's things to understand, but I can't say that, you know, I'm necessarily a great expert on all the latest evidence in gut biota with respect to ALS.
I mean, look, there has been a few publications to show that it may be changed, but what both Mehdi and I think would agree on is that really we should be looking at what the processes that happen in MND, and they're probably a better, you know, for a biomarker. For example, neurodegeneration, and P75 is involved in that. Immune activation, it occurs in MND, and excitotoxicity, which, you know, the measurements that Mehdi's doing, this is part of the process, rather than something that's quite far removed, which is the gut biota. It may change, but is it relevant? Is it relevant to what's happening in the neurons and what's happening in the body? I'm not sure.
We can perhaps return to that question next week as well, because Derek Stein, who is presenting next week, does a fair bit of work in this area as well. And also the comment that Duncan came up with also, that many patients do get frustrated with the long diagnostic process, which is obviously completely understandable. And a big advantage of an early diagnosis is people have a lot longer to accomplish some of those things they want to tick off before, while they can, basically a longer bucket list, as he put it. So that also is something to consider as well. So I'd just like to say thank you again to you guys for giving us a great overview of a very complex area, and two really diverse approaches to it as well. It's really interesting to hear the two different angles and how you can measure with sort of, I guess, two different ways, two different forms of technology. So thank you again to Mehdi and to Mary-Louise. And next week we'll have two more presenters talking about newer areas which are coming out as potential targets for developing treatments. It'll be the same time, same place, next Tuesday night on Zoom and Facebook. And all attendees will also receive a link to a recording of this, and it will also be up on Facebook in about a day or so. So please feel free to come back and watch it again, or distribute it to any friends, family, anyone else in your community you think might be interested in this. So I'll just say good night now and let you return to your evenings. And thank you for watching, and hopefully we'll see you again next week. Thank you all.
Thank you very much. Bye. Bye.